Next, I'd like to introduce Dr. Martin Brady. Dr. Brady is an electrical engineer by training. He's a co-founder of a company called Therataxis, and he has a very interesting background in image processing, massively parallel computing, neural networks, computer graphics, visualization. He's worked with Intel, also was assistant professor in a computer engineering lab at Penn State, and really looking forward to your presentation on pathways of flow and intraputamenal infusions. So, Dr. Brady, thanks a lot.
Thank you.
Nice to meet you.
So we've heard a lot of outstanding talks this morning and yesterday on convection-enhanced delivery, so it's a bit intimidating to follow up. Here I'm going to be looking, as an engineer, looking more from an engineering point of view rather than the biological perspective. More like a problem of controlling fluid flow through a poroelastic medium. There will be no math. However, there's a great deal of math underlying. Okay, so the research focuses on understanding the mechanics of extracellular fluid flow in the brain during convection-enhanced delivery. Our hope is with this understanding, we can make infusion distributions more predictable and more reproducible, something we've heard a lot about over the last several talks on CD. Knowledge of the physics governing infusions we think will be generally applicable to different regions of the brain. However, this study is dedicated to understanding specific issues related to optimizing infusions of the putamen. We assume that ideally you want to maximize the coverage of the putamen while minimizing loss to surrounding tissues. Depending on what you're infusing that, there might be variations on this, but that's going to be our starting assumption. So in particular here, I'm going to focus on the pathways that might lead to loss of infusing outside of the putamen. So our approach to understanding the physics governing putamen infusions is twofold. First, we've developed methods for measuring drug distribution and related tissue parameters intraoperatively in real time over the course of the infusion. We've applied these techniques in over 50 in vivo infusions into non-human primates to collect data for our experiments. The sort of measurements we get include calibrated 3D maps of drug concentrations over small time intervals, tissue expansion, infusion line pressure continuously monitored, tissue diffusivity, NRI geography, and other things. And then second, using this data, we construct mathematical models and implement them in software simulations that attempt to describe the observations. So this allows us to test and refine assumptions about the underlying mechanisms for our observed distributions. Valid models should ultimately lead to optimized infusion protocols and devices. So that's our hope. We can improve planning of infusions. We can improve the devices that are used for infusions using this knowledge. So the outline of talks to follow. First, I'll briefly go over the methods. Then we'll look at some of the major determinants of flow that we have investigated for putamen infusions. The complex elongated putamen geometry is one of the first problems you encounter that limits the amount of infusion from a single catheter before it begins to overflow the boundaries of the putamen. So this is endemic. So the next thing is that this is further complicated by the fact that there's a dynamic response of the tissue to the infusion pressure. It expands the extracellular space, this alters the flow, and complicates the understanding of where things will go. In addition to these sort of basic major factors, there are at least two significant preferred channels of flow within the putamen. One of them comes from the catheter itself, that's backflow, which we heard several others talk about, and that can infuse far from the intended source of the distribution. And in our experiments in the putamen, preferred flow also adjacent to the along arteries that are adjacent to the catheter tip can also be a major pathway for fluid flow, and it can infuse it outside the putamen as well. So I'll describe quantitative measurements characterizing the pathways, and then a bit on software simulations that attempt to model them. And finally, I'm going to conclude at the end with a few observations that attempt to extrapolate a little bit from the studies to the case of the human Putamen. So the data was collected from in vivo infusions, rhesus and cynomolgus Putamens. All was conducted at the University of Wisconsin. The majority of the infusions adhere to the protocol that we have there, 1 microliter per minute infusions, 2 millimolar solution of getateridol with BBS. Usually are bilateral infusions, 1 into each side. And we're continuously monitoring these infusions in the MR. Most of these infusions were done with a small, very thin step catheter, 0.36 millimeter OD, tip extending 3 millimeters from the catheter shaft. The shaft is 0.66 millimeters, so pretty thin. And most of the time these were closed, the end was closed with a fiber optic stylet. So the stylet wasn't providing stiffness, it just closes the end port during insertion and pull it back without letting in any air and infuse from the open end port. We did try some variations on this design, but usually with a catheter that's roughly these dimensions. All of these were done, all these catheters were placed intraocularly in the MR, so using this Navius Medtronic device, 1 on each side to place the catheter. So, there's more details on the targeting and the accuracy of that in the paper listed there. So in each of these cases, we did quite a bit of MR imaging for every case. A few weeks prior to infusion, we did some planning MRIs, and with those we gathered high-resolution DTI, 55 different angles. We did contrast MR angiography, very high resolution, high enough to see vessels in the putamen, and high-resolution T1 weighted imaging. And during the infusion, we continuously collect data. We're able to obtain the imaging required for T1 mapping, for quantitative T1 mapping at 10-minute intervals during each 100-minute infusion. And in addition, we were continuously monitoring line pressure during the infusion. The T1 mapping data that is used in analysis afterwards to produce concentration maps, again at 10-minute intervals. So we have full mapping in 3D of the concentrations. And from these concentration maps, we identify and categorize infusate inside and out the putamen. We quantify the total amount, that is the number of molecules of tracer gadolinium that we're seeing inside and outside the putamen and the different regions associated with different pathways. And we take all of this data and that's fed as input to our simulations. So this is an example of a well-behaved, best case infusion into monkey putamen, at 1 microliter per minute. The catheter was centered nicely. We had a nice round infusion sort of developing with low backflow. And the distance from the catheter to the nearest boundary is about 2 millimeters. So there's not much space in the putamen. That's about the best you can do. On the right, this image is one of the T1 mapping images, so it's a quantitative image of T1. And then overlaid on that, well, I've drawn in red, I've outlined the putamen, but then overlaid on that is the quantitative concentration map that we can compute. And this is the map at 10 minutes, the first one we added just after the start of the infusion. And you can see there's a scale on the right, the scale going from 1 millimolar per liter down to 0.05. 0.05 is the lowest, the least resolution where we could reliably extract concentration values. The reason why it doesn't go all the way up to 2 millimoles per liter, the infused concentrations, of course, because we're infusing into the extracellular space, as been described, which is only about 20% of the total volume. So you expect that your concentration that you observe is going to go down by a factor of about 20% from the infused, or about 0.4. Now, it isn't a uniform 0.4 across. We've heard some things about the expected uniform concentration distribution associated with the CED, and I'll talk a little bit more about why it isn't a smooth 0.4 across that in just a little bit. In the top left, then, this is a plot of measurements of our total concentration. So if you take and look at and integrate across the concentration map at all the points, sum all of that up, you can get the total number of molecules that you're seeing and that you're measuring. And the blue line is the total across the entire infusion, all of the infusate that I put in, how many molecules are there. And that's on top of, I know how many I put in, and it should be a linear plot. I'm infusing at a constant rate. And you can see we get a nice, very straight line. So the accuracy, precision at least, of this measurement is very high because we're integrating over a huge number of points. It not suffering from the noise of a single point. And we can calibrate that so it lies right on top of the amount infused. We can get a pretty accurate measurement of this. The red line is that portion of the infused molecules that are inside that putamen that I've delineated. So you can see how much is inside, and the difference between those two lines is how much we've lost, how much is outside the putamen. The third line in this graph is the green one. That's the pressure that we monitored throughout the infusion. So in the lower left, rather than amount or dose, we're looking at the volume distribution. And again, we're infusing into the extracellular space. So we expect that the volume of distribution will be about 5 times larger than the volume of the infusion. And again, the blue line is the total distribution, measuring everything that I can see. And it is, at the end of 100 microliter infusion, you have about 500 microliters covered everywhere. It's not all within the putamen. So the red line is the volume of coverage in the putamen. By the end, we've covered about 55% of the putamen with this one infusion. And looking at the top, the amount of measured traces outside is only about 21% because it's sort of clustered in the center. Okay, so that was our ideal case. That was the initial concentration map. That's at 10 minutes. And I can show what happens as 10-minute intervals or so, 9-minute intervals go on. You can see the new concentration maps on the right. And right around 35, 40 minutes, you can see that we're reaching the boundaries of the putamen. And we start to overflow, and there's no barrier to that. The infusate goes right past the boundary. So that's overflow. And in the graph in the top left, you can see that that's where we start to see this difference between the red and blue lines. You can see the leakage. Okay, so that was an ideal case. The catheter is centered the best I can do. This is only a slightly different case. The catheter is a little bit off-center. It's a little bit more medial. It's maybe a millimeter, half a millimeter from the nearest boundary rather than 2 millimeters from the nearest boundary. It's not greatly different. And again, this is our first 10-minute time point, 10 microliters in the infusion where we started. And if I run through this infusion, you can see that even at 20, 18 minutes, we're starting to overflow on the interior boundary now. And so the red and the blue, those lines diverge almost immediately, and we're overflowing from the beginning. And as a result, we've got more loss of diffusate from the putamen. And the interesting point is this is a very small change in catheter position, and fairly significant change in result, and how much is put into the putamen and how much is lost. So it tends to say that the dose is going to be very, very sensitive to catheter position. So the other thing that I want to step back to, besides catheter position matters, and the main takeaway from the slide, is this thing I mentioned earlier that, well, this is not a uniform-looking concentration that we might have expected. It's actually much higher near the catheter and falling off rapidly as we go out, and it's higher than that expected 0.2, 20% extracellular space we expect. At 20% or 0.4 millimoles per liter, that's about in the cyan or light blue sort of color. That's where we expect to see it. So it's much higher than that. There's a couple of ways this could happen. If you're infusing something that binds, it might bind and be held closer to the catheter tip or something that's retarded a large molecule, but that's not the case with this gadolinium tracer. The other possibility is that we get a perfectly uniform concentration infused in the extracellular space. The extracellular concentration is 2 millimoles per liter as put in. but it's the pore fraction that changes. We're getting expansion, and that appears to be the case. So normally, everybody knows that white matter expands. It expands easily with pressure far from the source, and that's usually what we assume, but it appears that gray matter can, in fact, expand. It doesn't expand for a long distance. It expands only under high pressure, only right near the catheter. But there is significant expansion when we're talking about at least infusions of a small focal site like this, and it should be taken into account. So we did corroborate this idea by looking at spin density images. So these are not proton density weighted, these are actually quantitative spin density. There's no weighting associated with T1 or T2, so there's no gap effect here. On the left side is the infusion we're looking at. In pre-infusion, you can see that the putamen is, the gray matter there is about the same as the gray matter elsewhere in the brain. And then post-infusion, you can see that there's an enhancement in signal all around where the infusion is. If you look down below, that's the concentration. That color image is the concentration map that I measured. And above, you can see that that's exactly where we kind of increase in spin density or increase in water. There's more water there. And if you look at the line, the yellow line going across horizontally, that's a single line of data where I've plotted those values in the far right graph. Green is pre-infusion, red is post-infusion. And you can see about how much excess proton density we're measuring, how high the signal is. It's about 25% higher right at the catheter. So that tends to tell us that there's more water there, tissue is being expanded, the extracellular space is pushing apart, there's more fluid, and that tracks nicely with this extracellular pore fraction that is predicted from this concentration enhancement. Okay, so what does this mean if I want to understand what's going on and model it? Most of our prior work, we assumed we were looking at infusions into white matter. We assume the white matter expands, it certainly does, and that's in the model, but the gray matter does not. And that's appropriate for large white matter infusions. If you look at this, if we do a simulation, well, I've got the same example, measured concentration in the center, this is what really happened. A simulation using our prior work, which is referenced below, does what you would have expected with convection enhanced delivery. We have a fairly uniform concentration across most of the infusion, and it's at about 0.4 millimoles per liter. It's also much larger, relatively larger infusion because rather than pushing tissue away, it's flowing out of the tissue. The other thing you see is that it appears to be flowing back along the catheter shaft more because since it can't flow out into tissue as easily, it's taking a path of least resistance off the catheter. So there's quite a few differences and implications that come from just that little bit of local expansion. So what to do about this? So this tissue expansion is a complicated thing to model because what we see is that what looks like near the catheter there's high pressure, very high pressure. The pressure falls rapidly away from the catheter, and along with this elevated pressure, you get tissue expansion, but the tissue expansion is proportional in some way to the pressure. So I need to modify the pore fraction to accommodate the pressure. But when the pore fraction changes, the conductivity of the tissue changes as well. So in order to understand what the pressure is, I need to know the conductivity of the tissue. The conductivity of the tissue is dependent on the pore fraction. The pore fraction changes with the pressure. It's a cycle. So I have to iterate. It's going to be a complex model. We did implement something like that, where we have a stiff gray matter, but it does expand with pore fraction, and the white matter does expand as well much easier. So that sort of model does appear to capture, if you look on the right, that's this modified model. That model does appear to capture pretty well what's going on. So that seems to be a pretty good, reasonable explanation for what we're seeing. Okay, so we have talked about elasticity, we talked about geometry of the putamen. Now let's look at the two pathways of highly directional flow. We've seen a lot about backflow already, and know about what the bad effects are. So these are some examples of backflow where backflow is let infusate leave the putamen. The mechanism obviously can carry infusate far from the intended path of flow. Here's an example we had, and here's what happens. In this case, this is, again, at 10 minutes at the first time point. You first see infusate, and it's already up the catheter shaft and out the top of the putamen. So this happens immediately. It's not a slow drift up the catheter. The first time point we measured, it's already gone 16 millimeters. It's already out of the putamen. And what happens from there? We don't continue to go upward. What happens is now we infuse outward from the catheter shaft out into the putamen and the white matter. Okay, so because we didn't lose infusate all the way out the top of the brain, it wasn't a total loss, we did infuse, and we do actually continue to infuse the putamen when it's not a total loss. About 30% of the infusate is within the putamen, pardon me, but about 70% out. So, pardon me. So it is a lot. You can continue to infuse even under these conditions. You can continue to get some result, but it's not nearly as efficient. And it's telling that there's a bigger spread of infusate actually right into the white matter than there is into the putamen, even though the pressure must be higher on the catheter tip. So there's lower pressure pushing this fluid out, but it's going farther. That tends to mean that there's much less resistance in the white matter. It's a less resistant material. We had several cases where we did have this backflow loss. We did get measurements. The one I just showed is the top blue line, where as a fraction, about 70% of the infusate was lost outside the top. And what you see is that we continue throughout the infusion. We initially are losing 30% or 70% of our infusate, and that amount continues. So we don't lose all of our infusate. We continue at this 30% is infused throughout the infusion. We had several cases where we didn't get backflow quite as far, just a few millimeters out the top. And that appears to be not quite as bad, but still, even when you're barely at the top of the putamen, we're losing about 30–40% of our infusate out the top. So a little bit of backflow, if it reaches beyond the putamen, is bad. A lot of backflow is much worse. From a modeling standpoint, there isn't anything new per se required to understand backflow that we didn't have in old models from our simulation. The additional work was done by Morrison in the 90s, and we refined that in the work listed down below. And the main issue is that with backflow, initially you have a small channel that is formed that pushes tissue away from the catheter. It doesn't take very much fluid to fill that because it's very, very thin, so it happens immediately. And then you've got this tension between fluid pressure that's being used to push away tissue from the catheter and fluid that's flowing out into the tissue. The easier it is to flow out into the tissue, the less backflow. The easier it is to push tissue away, that's shear modulus, the easier it is for long backflow. Once you get this channel established, you're back at a characteristic distance, then you tend to get the second phase where now we infuse, but we infuse not from the catheter tip, but from that entire pink channel, from everywhere with significant pressure. So the whole thing becomes a source. That's usually the case. Initial backflow, flow out from there, but there are caveats, so I'll show one here. This is a case where we had a nice, very low backflow in the lower left there as the infusion started, and then the animal shivered a little bit, and you saw motion blur in the MR, so we actually have evidence of that movement. And in the very next image, in the lower right, the backflow's changed. It's much larger, so that just a little bit of mechanical movement caused the backflow to rise. And things don't stabilize back down to where it was. Once that backflow is established, now we have a new distribution pattern. See in the upper right, we begin to infuse from the entire range of where the new backflow was. So motion, any mechanical motion is something to be avoided. Okay, and the other major pathway that we look for differential flow was along the blood vessels. This is a little volume rendering of monkey putamen. So you can see the putamens I was outlined in blue, and then I've got the MR angiography data just localized to the region of the putamen there. I don't know if it's bright enough to see, but down below from the middle cerebral artery, you've got these branches of lenticular striae, and they come up through the anterior portion of the putamen in the monkey, usually 4 branches and sort of curving diagonally and curving back as well. But they're covering a large portion of the region where you'd like to put a catheter. There are not a lot of places where you can put a catheter where you're not really pretty close to one of those. Then there are also anterior choroid arteries, and you can see little evidence of those in the back. So there's also a chance to run into those. We do see flow along both of these things depending on where catheters are placed. So this is an example of where we started off with what looked like a very, very good flow. This is at 10 minutes, so just at the start of the infusion, our first image. And in the top left, we're looking at the right side here. The left is a prior infusion. But on the right side, you can see the entire catheter. You can see a little bit of flow starting. The backflow is not too great. The catheter's in a good place. But if you look at the same time point down the bottom image, there's a sagittal view of this very same thing, same data set. And you can see there's a separate infusion ball down below, not around the catheter, in a separate location there. And it's already established right at the beginning. So we took and cut along this yellow line and did an oblique cut at 45 degrees, and that's what's right above. And now you can see the little top of the comma there is around the catheter. The catheter is sticking out of the screen diagonally 45 degrees. And a path that flows all the way outside the putamen. If I overlay the angiography on top, you can see that it's following one of the lenticular striae right down toward the mid-cerebral artery. So that looks like a pretty clear indication that we're losing fluid that way. So the same example, that same oblique cut, what happens is we start at 10 minutes, we've got this long path generated, and as the infusate proceeds, it looks actually a lot like what happened in the case of backflow. We have an initial characteristic distance that we flowed, but then we seem to flow out from that point. So we can lose a lot of fluid that way, and we start losing fluid right from the beginning. Okay, and is this significant? Well, in our case it was. So all these blue lines are the fractional loss of infusate at 30 minutes in 22 of our infusions from all sources, and the red is the amount we lost that I can attribute very clearly to one of these lenticular striae paths. And you can see that most of them had some measurable loss. I think 8 of them had losses of 10 to 35% of the infusion at this 30-minute time point that were attributable to one flow along one of these arteries. So how do you model this? Well, since it looks rather like backflow, we tried modeling it as backflow, basically. A characteristic distance of flow along the artery that happens to touch the catheter, and then it's a pressure source from there on out. And it looks like that's a pretty good model for what's happening. We need to adjust a little bit the pressures for the two sources. There's an interplay between catheter and artery, but this looks like the right way to think about this flow. So let me just very briefly touch on implications for human infusions that we might see from this. For our rhesus brains were about 700 microliter putamens on average, that's what I measured, from one side. So this is half measurement of putamen. We did some human volunteers, measured about 4,100 microliters. So about a factor of 6. Our cynomolgus was about a factor of 8 size difference in putamen. And I've put them side by side in there to scale. So it's hard to really truly measure width and height, but the striking thing is that while the human putamen may be a lot taller and it can tolerate backflow better, it's not really a whole lot wider. And so you're definitely you're at risk if you're a little bit off to the side. And we're very sensitive to catheter position and proximity to boundaries. You're definitely at risk of being a little bit off to the side, a little bit deep, and meeting a boundary earlier in this sort of infusion. So that's something to consider. I did a quick study of what if we were trying to fill the human putamen. This is just a simulation of the ideal, the best you could possibly do. And you'd want 800 to 1,000 microliters to fill a putamen that is 4,100 microliters. So I did an infusion for that long. And it looks like you can go about 100 to 200 microliters can be infused from a single catheter source, at the very best case, before you're reaching boundaries and you start to lose. So you can fill actually quite a bit of the human putamen, or you can get some coverage, but you'll also get a significant amount of loss if you're using one catheter. And just final thing, what about the vasculature? Is that even going to have any impact in the humans? Well, this is a volume rendering on the left, and the putamen's in blue, and MR angiography non-contrast for a human. And I hope it's visible there. You can see the middle cerebral artery. You can see the lenticular striates coming up to the putamen. They're very large, and they're right through the middle of the putamen where you want to go. And even in the T1, just plain T1 A and non-contrast, non-MR angiography, you can see the black dots. They're very large, and they look like they're going to be capable of having significant impact. So, summary. What we have is a very complicated, dynamic system which we need to model to understand convective flows. If you want to understand this detail, you really need to understand the underlying mechanisms. So this work is ongoing, but we think that that models of sufficient detail will be able to capture many of these observed behaviors, and thus aid in understanding the underlying mechanisms. We hope to apply these techniques to the design of protocols, plans, and devices for specific intraputamenal infusions. I thank all my collaborators, Agnes Foundation, Moses Funder, and as close collaborator throughout this work, Wisconsin, where all the animal work was done, Chris Ross, that designs medical devices, the pressure monitoring, and of course my colleague and co-founder, Brady at Texas, the expert in mathematical modeling of CD. Thanks.
Any questions for now? Great, I'm sure Mark will be available outside if you have any. I'm sure he'll be happy to pick them up. Thanks very much.